How?
Polyamine metabolism impacts T cell dysfunction in the oral mucosa of people living with HIV. HIV-1 infection causes TrEG/Th17 ratio skewing. Hey there, scientists. Today, we're diving into the fascinating world of HIV-1 infection and its impact on TrEG/Th17 ratio skewing. In a recent study, researchers explored the connection between polyamine metabolism and T-cell dysfunction in the oral mucosa of people living with HIV. Next, we postulated that HIV-induced TrEG dysfunction in PLWH4 might involve the polyamine pathway. We employed the oral lymphoid system of TCR-activated human tonsil organoid cultures, HTOC, in vitro 5. First, we examined ODC1 expression in different TCR-stimulated CD4+ subsets in non-polarized and uninfected HTOC. ODC1 is known to be upregulated in TCR-activated cells and highly expressed in vitro polarized Th1 and Th2 cells, but reduced in Th17 cells and TrEGs. 5 to 7. We compared CD25+ activated versus non-activated cells, IFN gamma plus Th1 cells, versus IFN neg non-Th1 cells, RR gamma T plus IL-17A plus Th17 cells versus RR TNEGL17N neg non-Th17 cells, CD4 plus CD25 plus FOXP3 plus TrEGs versus CD4 plus CD25 plus FOXP3 neg non-TrEGs, and CD4 plus CD25 plus FOXP3 plus PD1 plus IFN gamma plus TrEG-DIS, versus CD4 plus CD25 plus FOXP3 plus PD1 neg AFN neg non-TrEG-DIS, in TCR-stimulated HTOC. CD25 plus cells, Th1, Th17, and TrEGs expressed higher ODC1 levels compared to their counterparts, supplementary. Figure 5. Consistent to a recent study 5, Th1 cells showed increased ODC1 expression than TrEGs and Th17 cells. Our results also showed that TrEG-DIS expressed significantly lower ODC1 than non-TrEG-DIS. These data suggested distinct differences in polyamine metabolism between CD4 plus T cell subsets, supplementary. Figure 5, and provided a rationale for examining polyamines in CD4 plus T cells in the context of HIV infection. As shown previously, about 3 to 7% HIV GFP plus cells were observed upon HIV infection of these cultures. 418. HIV-1 infection causes significant depletion of IFN gamma and IL-17A plus effector Th subsets and results in the induction and expansion of dysfunctional PD1 plus IFN gamma plus FOXP3 plus cells, TrEG-DIS, 418. Because of the known role of Th17 cells in mucosal integrity, and the previous evidence of dysregulation of mucosal TrEG/Th17 ratios in acute SIV infections. 19, we examined TrEG/Th17 ratios in HIV-1 infected tox. As the markers we used distinguish the subsets with relevance to ODC1 expression, supplementary. Figure 5, we employed the same to identify Th17 cells in HTOC. To assess the effects of HIV and antiretroviral inhibitor, ARI, alone. In cultures, we had these control cultures to mimic HIV infection, followed by antiretroviral treatment in patients. Figure 1, we added ARI efavirenz 24 to 36H after HIV infection and allowed the cells to expand with ARI for 6 to 7 days. Similar to our previous findings. 418. TrEGs showed significant cell death and were restored with ARI efavirenz, figure 2a, top. After confirming TrEG-DIS expansion during HIV infection that persisted in the presence of ARI, figure 2a, bottom. We assessed the frequency of Th17 cells by determining the expression of RR gamma T, CCR6, and IL-17A in CD4 plus T cells. The cell numbers of Th subsets were also determined in these cultures. Supplementary Figure 6a. Our results showed that HIV-1 infection led to a significant loss of Th17 cells and the ARI treatment post. HIV infection was not able to restore them in vitro, Figure 2b, c. Alterations in the frequencies of these two subsets resulted in significant enhancement of TrEG/Th17 ratios during HIV infection and were sustained even after the ARI treatment. Figure 2d. These results prompted us to determine the role of polyamines during HIV infection in the presence of ARI. Now, let's break it down. HIV-1 infection causes significant depletion of IFN gamma and IL-17A plus effector Th subsets, leading to the induction and expansion of dysfunctional PD-1 plus IFN gamma plus FOXP3 plus cells, also known as TrEG-Dys. This imbalance in TrEG/Th17 ratios can have a profound effect on mucosal integrity, which is crucial for overall health. To understand the underlying mechanisms, the researchers examined the oral lymphoid system of TCR-activated human tonsil organoid cultures, HTOC, in vitro. They found that HIV-1 infection led to a significant loss of Th17 cells, and even treatment with antiretroviral inhibitors was not able to restore them in vitro. This resulted in a significant enhancement of TrEG-Dys/Th17 ratios during HIV infection, which persisted even after treatment. So, what's the role of polyamines in all of this? Well, the study revealed distinct differences in polyamine metabolism between CD4 plus T cell subsets. ODC1, an enzyme involved in polyamine synthesis, was found to be upregulated in CD25 plus cells, Th1, Th17, and TrEGs, but interestingly, TrEG-Dys expressed significantly lower levels of ODC1 compared to non-TrEG-Dys. This sheds light on the potential impact of polyamines on T cell functions in mucosae. The researchers also delved into global metabolomic profiling of saliva and transcriptional profiling of the oral gingival mucosa. This comprehensive approach uncovered metabolic alterations in people living with HIV, including dysregulation of arginine and polyamine pathways. The upregulation of key enzymes involved in polyamine synthesis, such as spermidine/spermine N1, acetyltransferase, SAT1, and ornithine decarboxylase, ODC1, suggested increased polyamine synthesis in the oral mucosal microenvironment of PLWH. This study provides valuable insights into the intricate interplay between HIV infection, polyamine metabolism, and T-cell dysfunction in the oral mucosa. It opens up new avenues for further research and potential therapeutic interventions to restore TrEG/Th17 balance in people living with HIV. And there you have it, scientists. A closer look at the fascinating connection between HIV-1 infection, polyamine metabolism, and T-cell dysfunction in the oral mucosa. Stay tuned for more exciting updates in the world of scientific research.
Pharma Pal.